Hello, everyone, and welcome to today's webinar, Achieving Clinical Quality for Human iPSC Generation and Expansion. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Miltenyi Biotec. To learn more, visit miltenyibiotec.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speakers, Dr. Boris Greber, Head of R&D, iPSC, CT, Catalent Cell and Gene Therapy, and Dr. Chao Sheng, Product Manager, Regenerative Medicine, Miltenyi Biotec. Boris and Chao, you may now begin your presentation.
Yeah, so thank you very much for the introduction. I'm happy to talk about our ongoing activities at Catalent around the iPSCs, their generation and manipulation. So to start with, I would just like to give a brief intro into the iPSC add-on to Catalent. So this was a recent acquisition, basically done last year. The original company was called RheinCell, located here in the Cologne, Düsseldorf metropolitan area, and our aim here was to generate a clinical grade universal starting material, so IPS cells under GMP conditions, to form the basis for future workflows in cell therapy. And now being part of Catalent, I think this really creates new opportunities, not only for our side here, but also vice versa, to really harness the full potential of these cells. So, for example, we have extensive manufacturing capabilities now within Catalent at Houston, as well as at our site in Belgium, in Gosselies, even for commercial scale cell therapy manufacturing beyond the phase 3 clinical trials. And in turn, I think we can really profit now from the resources that there are at Catalent. For example, plasmid manufacturing at GMP quality, as well as other modalities, which can now serve as reagents in GMP workflows using iPSC. So we really hope to create these kind of synergies at Catalent now. So what I would like to focus on now is the manufacturing workflow. So as we have set it up with the iPSCs, and also highlight the pipeline for quality control that we've set up, and also come to talk about the starting material that we use here in the manufacturing process. And later on, I will talk about more current and ongoing activities in terms of differentiation of the iPSCs and development of future workflows, and also come to talk about the overall strategy for reprogramming. So the manufacturing workflow is based on a rather simple workflow using episomal reprogramming. We try to keep things simple and robust here, really, to make sure that reprogramming experiments workflows really work every time we perform them. And this is all done with the IPS Blue medium from Miltenyi. At the time, I can say it was one of two media on the market, GMP grade iPSC media on the market, and we compared these against each other and simply found that the IPS Blue medium was superior. So we stick to that and build a workflow based on that medium. Otherwise, we are using xeno-free culture conditions, chemically defined culture conditions, as you would expect from a GMP workflow. And like I said, keep things really actually quite simple and manual. So the workflow starts with import of hematopoietic stem cells from a collaborating cord blood facility. So this is a GMP grade starting material, and we then introduce this into our clean room facility here in the building to then subject cells to reprogramming, and this will give rise to single lines, single colonies that are being isolated and grown up to independent cell lines. So we are establishing several cell lines for each donor, and we're then making a quality assessment and proceed with one selected clones while we freeze stocks of these candidate clones, so we keep them for potential later use. But this one selected cell line is then grown up, and until the vectors that are used for reprogramming are fully eliminated, and finally the cells are expanded and cryopreserved. And all the QC is obviously taking place, or most of the QC is taking place then outside the clean room facility in dedicated QC labs. So the QC assays can be subdivided into different categories, if you will, into safety-related assays, into assays looking at genomic integrity of the cells, and into assays that address cell type-specific characteristics, so differentiation potential of the iPS cells, for example. So we're trying to address all these important points using a panel of assays, and I would just like to highlight a few examples. So this is showing you in process controls that are carried out in the clean room. So we're taking samples at regular intervals and really use stringent Tacman-based assays to verify that the vectors are becoming eliminated over time in the cells, and only if we observe that repeatedly over several passages, we would switch over to amplifying the cells and cryopreserve them. Another important issue is the genomic integrity, like I mentioned. So we're doing, on the one hand, classical karyotyping, following international standards for tumor diagnostics. So this involves numerical analysis as well as structural ones, using multiple metaphase spreads. But in addition to that, we're also using full genomic sequencing to assess the mutation load in these cells and to rule out that there are functional mutations in cancer-associated genes, for example. So on the right-hand side, you see an analysis in this regard, where we also revealed that the mutation load in our lines tends to be rather low. So you see the four data points on the right-hand side in comparison to iPS cells derived from adult starting material. So we could really trace this down to the starting material, which is, I think, implies really a key advantage of here using cord blood. Yeah, and with regards to the specific properties of the iPS cells, we're certainly assessing differentiation potential in several ways. On the one hand, of course, in the GMP QC workflow, where we mostly look at semi-spontaneous differentiation, but we're also developing, and our clients are testing cells for directed differentiation capability. So we have generated endothelial cells from these lines, as well as pure RPE cells with a new protocol that we've developed, and we've also subjected these cells to cardiac induction at high efficiency in all cases. So we're quite happy with the properties of these cells indeed. Just briefly to summarize the points about the starting material that I already mentioned in part. So we think that cord blood is a preferred starting material for reprogramming iPS cells for clinical use. First of all, because it's a clinical material by itself. It's used for therapeutic purposes, and as such, it's extensively pre-tested for viral contaminations, for example. So we're building on that material, and another biological argument is really the the fact that these cells are neonatal, so the mutation load in that material is really low, and that translates into the iPS cells, as I tried to indicate. Now, in the final part, I would just like to highlight a few workflows that we're working on. So we are now building on what we have basically on the iPSCs. We would like to establish differentiation protocols and GMP compatible workflows on that basis. And I would also like to highlight the overall strategy we use for generating the iPS cells. And I'd like to start with that aspect. So as you know, when setting up an allogeneic approach based on iPSCs, you will face the issue of immune rejection, simply because donor and recipient are not identical, and the cells are easily becoming rejected in the patient under normal circumstances. So there are approaches needed really to address that issue, because immune suppression, which could of course be applied, but always comes with side effects, quite severe side effects actually. So our strategy to address this issue is to use HLA homozygous donors as a starting material. So these are it's a rare condition in the population to be HLA homozygous. This occurs at really low frequency, and we are, so to speak, cherry picking our cord blood bank for these privileged individuals. Because using HLA homozygous cells, as indicated here with these color codes, really simplifies the challenge of immune matching. So it's much easier to find matching recipient patients using HLA homozygous cells, because you only need to consider one of the two parental HLA alleles for each gene, as shown here, or indicated here with these color codes. Of course, I should also mention complementary strategies to achieve immune compatibility, and gene editing is the most prominent one in this regard. So you can think of manipulating HLA genes as well as other genes to make the cells immune compatible to basically any potential recipient in the population. And there are also hybrid approaches where you're using HLA homozygous cells as a starting source, and then do a partial deletion of the HLA genes, for example, so to enhance their immune compatibility. And so I think these different approaches, they're definitely not competing, they're complementary. And in fact, we think that the HLA homozygous iPS cells that we've generated would also present a great starting material for enhancing the applicability of these lines by gene editing. So, but again, discussing the homozygous HLA homozygous approach. So these are some calculations that we performed, where basically compatibility of the cells to the population is plotted against the individual genotypes, the HLA haplotypes, and these are ranked here according to frequency in the population. So you see the most frequent HLA haplotypes on the left of the left-hand chart, and you can infer from such calculations that you just need a handful of, or maybe 10 cell lines or so to, in our case, to basically match every second patient in the population, which I think would be great. Like I said, gene editing can also help to increase the matchability in the population, and also obtain access to other populations. Yeah, the overall applicability of these cell lines could be enhanced this way. On the right-hand side, you see the current status where we are. So we see that we basically have access to the most frequent haplotypes, making this strategy, I think, a viable one. But we've also learned really over the last years that other things play a role as well. And an important point here is really donor eligibility, so qualification of donors. In terms of regulatory requirements in the US or in the European Union, as well as in Japan, for example. So we're trying to really explicitly match these requirements with new cell lines. So for the existing cell lines, what we had to do is basically take what we have, because we're using the HLA homozygous approach. So we need to use really existing material for that, and for that reason we had to develop mitigation strategies here for these existing cell lines, comprehensive ones, strong ones, we think, but like I said, as a complementary strategy, I think we would also now like to really fully fulfill these requirements from the start with new cell lines. So then just a few more slides on the current differentiation activities. So we are working to establish efficient, improved protocols that could form the basis for future GMP workflows for iPS cell differentiation. So generation of interesting cell types of clinical importance. So the MSCs are one example. We've developed a simple one-factor-based protocol here, very robust and fast also. The MSC-like cells that we generate express typical MSC markers and show extensive proliferation potential. As an example, so we hope to be able to generate cell banks of these MSCs soon to be able to offer them to potential partners and clients. Another example is immune cell differentiation. So we acknowledge really the growing interest in cancer immunotherapy based on iPS cells, and we think that the existing protocols are not really perfect. Perhaps we definitely have potential for further improvement. And what we're doing here is we are trying to really optimize also the early stages of differentiation. So the initial induction of hematopoietic stem cells, or even earlier, the induction of hemogenic endothelium. So the precursor really of these hematopoietic stem cells, and the data shows here that on day 7 we indeed have a cell type that is in line with the idea that these cells are hemogenic endothelium, so CD34 positive, 45 negative, and positive for endothelial markers like CD144. But then upon further treatment with a corresponding cocktail of molecules, this phenotype switches to a CD144 negative, CD45 positive. So in line with a switch to an HSC identity, and we then take these cells, which are naturally 3D cells, suspension cells. So we then differentiate these cells further in 3D format into NK cells, for example. And just another quick slide here on the genetic engineering activities at Catalent. So we are currently building workflows for gene editing, for knocking out genes at high efficiency, but also for knocking in genes into specific sites in the genome. We're trying again to build up efficient and robust, reliable workflows that we're then later on able to use for our own internal aims, but also to offer it to partners and clients outside Catalent. So the data here shows you the double knockout of HLA class 1 and class 2 in the context of creating so-called universal iPS cells. And it shows you that we've successfully isolated homozygous double knockout clones, and the FACS data here on the right-hand side basically validates that. So the wild-type iPS cells, upon stimulation with interferon gamma, induce HLA class 1, but the knockout clones obviously fail to do so. And likewise with HLA class 2, in this case, expression in the iPS cell ground state was very minimal. So we differentiated the cells into endothelial cells according to our new protocol, and then at least we obtained partial induction of HLA class 2, which I think was sufficient to show that the knockout clones are indeed deficient of HLA class 2. So we're quite optimistic that this presents a good basis for GMP workflow or GMP-like workflow in the future. So just to briefly summarize, we have iPS cells generated under GMP conditions. We are offering these iPS cells to license and form a basis for future clinical workflows. At the same time, we're also offering basically the workflows themselves as we have set them up, so custom reprogramming or QC services around the core workflow. And the current activities are really focusing more and more on differentiation protocols and GMP compliant workflows based on these protocols, as well as on gene editing. So we hope to be able to offer in the future really attractive protocols and workflows to partners, and we actually right now also partnering at the development stage. Yeah, so thank you very much for your attention. I hope this was interesting, and I'm happy to take questions.
Well, many thanks, Boris, for the great talk. And I think the workflows that you just presented on GMP-compliant human iPSC generation, expansion, and the quality control analysis at Catalent cell and gene therapy set great examples in the field. I think all of us, we can learn a lot from your work. And also, thanks for the nice introduction in the beginning. So my name is Chao Sheng, and I am the Global Product Manager for Regenerative Medicine Manufacturing at Miltenyi Biotec. So, and now, actually, I would also very quickly introduce the established workflows at Miltenyi Biotec for automated, closed, and GMP-compliant manufacturing of human pluripotent stem cells. First, very briefly, a few key facts about Miltenyi Biotec. So the company was founded in 1989 and currently has more than 3,500 employees worldwide. And among them, 25% are located in our global R&D department, who are constantly advancing our knowledge in various fields, including regenerative medicine. Well, speaking of manufacturing of PSCs and their derivatives for clinical applications, so we all know that they are very challenging and complex endeavors, and most of the current manufacturing processes involve multiple very expensive instruments, very skilled operators, and high-class clean rooms. And also many separated open handling steps are also introduced in these workflows. Meanwhile, standard in-process control and quality control assays are required throughout the entire processes. In addition, well, in certain applications, the desired cell populations also need to be isolated in a sterile, gentle, and GMP-compliant environment. So all these factors together could really limit the manufacturing skill, compromise the reproducibility, at the same time, drive up the manufacturing cost. So how to make these very complex cell manufacturing processes standardized, scalable, and also more affordable for the end users? And here, there are some solutions from Miltenyi Biotec. So with the Climax Prodigy, the separated open handling steps, multiple devices, skilled operators, and high-class clean rooms, they can be integrated into one automated and closed system. And the users could build customized PSC expansion and differentiation processes with the Climax Prodigy. With the MaxQuant analyzer, then standardized iPSC QC assays can be realized by automated flow cytometry analysis. And finally, the MaxQuant title cell sorter can be applied to isolate the desired cell populations with a gentle, fast, and GMP-compliant cell sorting technology. And today, actually, we will focus on the Climax Prodigy. So together with the single-use tubing sets, the Climax Prodigy allows automated cell processing from starting materials to final products. Upon your request, it can automate all these different handling steps, including sample preparation, dense-degraded centrifugation, cell washing, etc., until final product formulation. And based on the Climax Prodigy platform, we have also developed a broad range of software applications for various cell types, for example, for CAR T cells. And for the adherent cell types, such as PSCs and their derivatives, so we have developed the adherent cell culture process. Using the Climax Prodigy adherent cell culture process, cell cultivation can take place in two different units, the centrical unit and the external culture vessels. So first, there's a Climax Prodigy chamber lies inside the centrical unit. It supports automated adherent and non-adherent cell cultivation. So the centrical unit also supports controls of the temperature and the concentrations of air and CO2. So this Climax Prodigy chamber offers 100 square centimeter surface space. Well, if larger surface space is required, then external culture vessels can also be easily connected to the Climax Prodigy tubing set via serial welding. For example, these multi-layer cell stacks. So the users can connect maximally 3 of these 5-layer cell stacks at the same time. This will offer roughly 10,000 square centimeter surface space. So actually, when we designed this software, so our goal is actually to make sure that it is flexible enough to culture different cell types. So that is why we modularized the individual cell handling steps. Now you can find these different modules on the main menu for density gradient centrifugation, for inoculation, for coating, medium change, harvest, etc. And the users can freely select and combine these different modules. As a result, very complex workflows, such as directed differentiation of PSCs, can be realized on the Climax Prodigy adherent cell culture process. Within each module, the users can also set up the parameters according to their established SOPs. For example, in a coating module, the user can set up the coating temperature, coating time, the volume of the coating reagents, all according to their established SOPs. So we have established the workflows for GMP-compliant expansion of human MSCs and PSCs, as well as directed differentiation of PSCs into midbrain dopaminergic progenitors and cardiomyocytes. So next, we will quickly show you some results on GMP-compliant PSC expansion using the Climax Prodigy. So for this workflow, we have first developed the IPS BlueGMP medium, which is manufactured and tested under the quality management system ISO 13485 and is designed following the recommendations of USP 1043 on ancillary materials. So it is currently available in bottles, but very soon we will also offer the medium in bag format. So we just saw in Boris's talk where they used this medium for GMP-compliant iPSC manufacturing. So in our experiment, the result shows that this medium enables very stable and efficient human PSC expansion as clusters or as single cells. So it also supports fast culture reinitiation after cryopreservation. And it is based on actually the same formulation as our research-grade StemX IPS Blue xenoframe medium, thus can support seamless transitions from research to GMP-compliant processes. Then, by applying this IPS BlueGMP medium on the closed and automated Climax Prodigy adherent cell culture process, the users now can choose and combine these different modules to automate all these complex handling steps. For example, coating, PSC inoculation, medium change, and harvesting, etc. We have also performed side-by-side comparisons between manual and Climax Prodigy-based PSC expansion using our reaffinity recombinant antibodies and the StemX trilineage differentiation kit. And looking at the expression levels of the potency markers and the trilineage differentiation potentials, so these two groups, the Climax Prodigy versus manual control, so they really give rise to very comparable results. Then by using the centrical unit and the different sizes of external culture vessels, the users can also easily reach different PSC expansion scales on the Climax Prodigy with much less open handling steps in comparison to the manual process. We know that establishing GMP-compliant PSC banks is a very important step in many clinical applications. So for this purpose, the closed and automated Climax Prodigy human cell culture process, together with the IPS BlueGMP medium and our QC reagents, so they could be very valuable assets. Here, just a very quick example. So recently, Dr. Raphael Kroll from the Saira Foundation in Japan has successfully transferred their manual clinical grade IPS expansion workflow into the Climax Prodigy adherent cell culture process. So he has already presented his work at the ISCR 2021 Tokyo Symposium and also at a joint webinar with us last year. Well, actually, that's already all from my side today. So this is the disclaimer of my presentation. So thanks a lot for your attention. I'm very much looking forward to discussing with you in the Q&A session. Well, if you are interested in any individual meetings to discuss how to adapt your established manual process on the Climax Prodigy, so please contact me via email at chaos@miltenyi.com. Thanks a lot, and looking forward to discussing with you later. Thanks a lot.
Thank you, Boris and Chao, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, will the new IPS BlueGMP medium in bags have the same formulation as the current medium?
Well, I can maybe quickly comment on that question. So, I mean, the short answer is yes. So the formulation will stay the same. So the only thing is that, in this new bag format, that the cytokine TGF-beta1 will be already included in the supplement, so that the users, they do not need to add this cytokine additionally.
All right, thank you. Next question. Will the Climax Prodigy affect the pluripotency properties of PSCs during the automated expansion process?
Well, I mean, I think this is, maybe I can also quickly comment on this. I think this is a very, very important question. So, for the manufacturing, then, quality is really the key factor. So, as I think briefly mentioned in my presentation, so the PSCs that we expanded using the Climax Prodigy adherent cell culture process, so they show actually very high expression levels of these pluripotency markers. They include the TRI-160, SSEA4, SSE5, OCT4, SOX2. So, and they are also able to differentiate into all the three germ layers upon differentiation. And at the same time, so when we compare the cells that are expanded manually, and we see that these two groups, that they are very comparable in terms of the pluripotency marker expression and the tri-lineage differentiation potentials. So that, based on these experiments, so we believe that, the PSCs that they maintain their pluripotency properties during the automated expansion in the closed Climax Prodigy platform.
All right. Our next question is, do you have medium for serum fran human myoblast expansion?
Well, I believe maybe I can just quickly comment. So I think I need to double check this. So I mean, if I find more information, then I could, maybe answer you via email, or quickly we can also discuss this via email.
All right, thank you. Next question. Does the Climax enable any in-process monitoring of cell media such as pH, DO, glucose, or lactate?
Well, At the moment, what we could do on the Climax Prodigy when we manufacture the cells is that we have the sampling pouches, so that every time when you passage the cells, that you could collect samples. Then then you could measure these factors you mentioned, like pH, DO, glucose, lactate, etc., in the collected sample. So this is at the moment how we can achieve this. But of course, we are working on how to improve to have a more precise in-process monitoring of during the cell manufacturing.
All right, thank you. Next question. Apart from the complementarity of different cell lines in terms of HLA haplotypes, do your cell lines differ from one another in other, e.g. differentiation or properties?
Yeah, that's a question that goes to me. Yeah, I mean, of course they do differ to some extent. I mean, the cell lines, if you will, they are individuals, right? They are isolated from different people, and we've seen many times that the genetic background is really the main determinant of cell line properties. In principle, if you use a stringent protocol for differentiation, that should actually make no difference between cell lines, but there are good protocols, I would say, and less stringent ones, and the less stringent differentiation protocols will certainly also reveal these kind of differences. Yeah, overall, we've shown the cell lines are really pluripotent, but there may be differences in differentiation capacity depending on which protocol you're using.
All right, thank you. Next question. You highlighted a lower mutation load in your iPSCs as compared to other lines. I did not quite understand how you could put that down to the starting material. Could you please explain this a little more?
Yeah, so what we've done is we made a mutation load comparison between our cells and published cells, and what you can do there is really also look into the frequency at which these mutations occur in the cell population. And what you normally assume is that if a mutation was present in the starting material, then it should be present in the cell population that you analyze by either 50% as a heterozygous or 100% as a homozygous mutation. And if you use that as a criterion, you can then distinguish between mutations that have been present already in the founder cell of a IPS line and mutations that have only arisen later. So using that, we could then boil these differences down to differences in the starting material rather than differences in the manufacturing process. So it's really the cord blood source we think that makes the difference here.
All right, thank you. Next question. What is the cell count capacity for the current Prodigy platform?
Well, maybe I can also quickly comment on that question. So, I mean, this is really depends. So, what cell types that you are culturing with the Climax Prodigy platform. So, as I showed in our case, this time with about the pluripotent stem cells. So, using this adherent cell culture process, that you can actually connect the external culture vessels, like the cell stacks, to really enlarge the surface space of your cell cultivations. So then that means that the total cell number, the scale, it also depends on the what kind of cell culture vessel that you are using for your process. For example, if you're using the chamber, which is already part of the tubing set, so this chamber has 100 square centimeter. So that if you culture the PSCs, so we normally inoculate around 1 million iPSCs, and then upon the once they get confluent, then when you harvest the cells, normally you could reach roughly 5 times 10 to the 7 pluripotent stem cells in the chamber. But when you further expand it to, for example, 1-layer cell stacks, maybe then you could reach like 2 times 10 to the 8. So 200 million PSCs. And then if you use the 5-layer cell stack, which has much bigger surface space, then when you harvest the cells, then you could probably reach already 1 billion 1 times 10 to the 9 pluripotent stem cells when you harvest the cells. But of course, this depends. We know that there's very some differences between different PSC cell lines. So that also depends on the cell line, how many cells you can harvest and how many the cell numbers that you can harvest at the end of your process.
Do you want, Chao, do you have any final comments for our audience?
Well, I mean, like I mentioned, so I mean, if I just a quick comment from my side in the end. So, I mean, many thanks to LabRoots, and also we thank a lot to Boris, for his very nice and very informative presentation, showcasing the GMP compliant iPSC manufacturing platform at the Catalent cell and gene therapy. So, I mean, I think we all share the same excitement in the field because we see that there so many scientists right now are trying to translate their research expertise, their experiences into clinical applications for the treatment of some devastating diseases. So, and we are also very happy that our products, our technologies, could support the customers in their processes. So. Just as Boris mentioned, so for example, the iPSC BlueGMP medium could be a great asset for for GMP compliant expansion manufacturing of human pluripotent stem cells. So I mean, if you have if you're interested in these technologies from us, like this medium or like the Climax Prodigy, the cell manufacturing platform, so then we could also discuss this in a individual meeting via email. So my email address is very easy to remember. It's chaos@miltenyi.com. So actually, Michelle, I think there's one more question.
Yes, one question just came back in. Given that a derivation of few iPSC clones from one patient costs around $10, how high cost?
10,000.
Is it 10 or 10,000? Okay, I saw the period and I was thinking, how high cost would the cost be under GMP conditions, comparing also cost today and envisioned in a couple of years when the technologies become more routine?
Well, I think this is definitely a great question from Daniel, but I think maybe Boris can have a much more informative answer to this specific question. Yeah, because I think that the cost or the cost of good for the manufacturing of the GMP compliant iPSC lines, like I mean, I think global wise is around 10 to 15,000 US dollars, but it's decided by manufacturers, and it probably is also vastly different between different countries.
All right. Well, then we will leave that question for Boris to address via email. Well, thank you again, Boris and Chao, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Miltenyi Biotec, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.